Good morning, I'm Wang Yici, and now I am pursuing a master degree in life science department. In life science department, we have many basic research about animal and plants. And I'm now studying in the field of plant because I love growing plants. Since I was in undergraduate school, I learned many skills of planting fruits, vegetables, and flowers. But there is something always bother me a lot is that plants sometimes got infected or attacked by the pathogen or insect, and I usually need to use pesticide or the plant will die. And most of the time, plants will be attacked by the pathogen, and they will easily to die, and the pathogen will be stronger. But here I have find a plant that it acts the opposite way because this plant can utilize the pathogen, and the plant will be stronger after infected by the pathogen. This plant is called Tianma, and I want to understand why this plant can conquer the pathogen and utilize the nutrients. And how does this plant look like? It looks like a potato. But unlike a potato, this plant is very expensive. Imagine you are buying a package of potato in the supermarket, but this package of potato costs about 2,000 NT dollars. So this is the price of Tianma. It's very expensive. As a Chinese herbal medicine, it is used for relieve the neural diseases such as dementia and Huntington's disease. So many want to grow this kind of plant. However, it's extremely hard to grow. So one reason why this plant is so hard to grow is that it doesn't have the leaf and roots. Means it cannot photosynthesize to gain nutrients, and also it doesn't have roots, so it cannot obtain any nutrient from other way. So there's a mushroom called honey mushroom. It will associate with this plant, and with this honey mushroom, this plant can grow to the size of the potato. Without the honey mushroom, the plant can only grow like a rice grain size. So this mushroom is very essential to Tianma. But however, the mushroom is actually a wood decay fungus for other plants. It will let many trees collapse and die. So it seems like Tianma is in the top of the ecological pyramids. And I'm wondering why can the Tianma conquer the honey mushroom without dying? I think there must be some changes in the Tianma. So for my research, I want to see how the Tianma change after infection by the honey mushroom. Since cell wall is the first barrier to face pathogens, so I will focus on the cell wall changes in the Tianma. So I think maybe there are some changes in the cell wall that can defend the honey mushroom. So for this is my outline, and I will show how I collect the sample next, and I will cut the sample into some slices and observe some special type of cell. And I want to verify the cell, the change of the cell. So I use two methods to see, and one is for the localization of the cell wall protein, and one is for the expression of the cell wall protein. And then I will find protein is specific to the cell wall for the remodeling. And this is the sample of Tianma. That I mentioned, Tianma can grow to the potato size, but it takes a lot of time to grow. So I just compared the Tianma before and after infected by the honey mushroom. And the left one is the Tianma without mushroom, so it's about 1 centimeter long. But after infected by the honey mushroom, the Tianma will grow to the size about 5 times bigger. So I will like to do some section to see the Tianma tissue looks like what. But before doing the section, I will define what direction I do the section. I will do cross section. Cross section is like here. This one is cross section, and the left one is like a longitudinal section. So I will cut Tianma into this size cross section. And these are the two Tianma, the uninfected one and the infected one. And after doing the cross section, I can find that the infected one have formed a very thick layer, just like a cell, like a wall of the boundary. And take a look about the real section here. We can see the cell become very large in the infected one. And I think maybe there are some changes in the very large cell. So I will focus on the large cell forming. And in previous study, we know that there's a group of protein related to the cell wall remodeling. And I have picked up 2 candidate protein. One is extensine and one is pectin methyltransferase, and those are related to the cell wall formation. To see the cell wall protein, to see if there are any cell wall protein in the large cell layer, I use 2 methods. And the first one is called immunohistochemistry. This way is to use the special tools to detect any specific things. So imagine here is the many stationery here. And can you help me to find out the red paper clip? And you may think it is very difficult to find in a short time. So now if we have a magnet, we can easily find the red paper clip. So immunohistochemistry is the same way. I can use antibody to detect the protein I want to see. So by this way, I will know the antibody will bind to the large cell if they have the candidate proteins I want to see. So, and beside observing the location of the protein, I also want some neural data for knowing the protein expression. So I use the real-time PCR to detect. And before using the real-time PCR, the sample collection may be different. I also cut the sample into very thin slices, and then I use the laser to cut the specific cell I want to see. And here you can see the red line I just draw on the cell, and then use the laser will follow the line to cut down this cell. And then I collect all the large cell, about 1,000 cells, and then for RNA extraction, and do the real-time PCR. And real-time PCR is like if you have a book you want to print for many copies, and the real-time PCR can copy a lot of the candidate thing you want, and by knowing that how many copies it will produce means how many candidate genes is in original. So here I have some results about the immunohistochemistry. You can first look at the right hand side here. The section with the label of the fluorescent, and you can see some some cell is bright. And I put this picture into the gray one to compare their difference. And the left one is the uninfected TMR section, and the right one is the infected TMR section. And I can see that in the infected TMR section, there are some bright thing in the cell. Means there are many labeling protein in this cell. So, and this label is for the extensing. The extensing is the function of forming new cell wall. So from this result, I can know that maybe there are some protein related to the cell wall thickening, because they form a lot of cell wall in this case. But also, if the cell wall, if TMR also need some nutrients, they will make their cell wall loosen, and that the bloodstream should come up and have some nutrient from it. So using the cell wall loose or tight can make the TMR to control whether it will gain nutrients or defense. The adjustment of loosening or tight of the cell wall is essential to TMR to control the fungus. So how in the future we can fully understand about the cell wall remodeling mechanism in TMR, and then we can apply this function of pathway in other plants to avoid pathogen invasions. So this is my research. Thank you for listening.